Katy and Emma would like to get going. Okay, Katy and Emma.
Okay. This works? It does. Great. Okay, should we keep this? Okay.
Are they on?
I think so.
Test it? Yeah. Okay.
Yeah.
Welcome, everybody. It's really good to see you all here. It's a great pleasure to moderate this session with Emma, which I just met through this panel, which is really also a wonderful opportunity to then compare notes across continents, across disciplines. And we also invited 3 brief presentations. So these presenters, if they want to sit here in the front, that would be marvelous. This way they can see each other's presentations. And then later on, the 5 of us will sit in these nice couches here, and you can ask us all kinds of questions. So please get your questions ready. I think Emma will start,
yes.
And then I have a brief comment also to make, and then we go over to these very brief expert presentations.
Yes, okay, so I will start by setting a bit the scene for this session. The title is Strengthening Integration Pipelines. This is our very long blurb that we just slapped in a slide, but in brief, what we want to cover in this session is a bit more maybe of an operational view of what building an integration pipeline looks like and what features do we want to essentially infuse in our integration pipelines. And we'll do that with a bit of setting the scene on some questions that we thought about when we were essentially given this task to, start this session. And then we'll look with Katy's work first and some of the flash talks at a bit some more like concrete examples of integration pipelines. And I'll just start by setting the scene. One of the first things that we thought about when we were thinking about integration and integration pipelines is what are the features of a human cell atlas that we'd like to essentially have and that we'd like to use. And the first feature that we could think of was, of course, that we want the atlas to be comprehensive and comprehensive in terms of many different axes. So there will be one of the axes that we can think of in terms of samples that we cover. Here, for example, I actually took data from the CELLxGENE collections and just plotted the number of healthy data from how many healthy donors do we have data for different tissues. And you can see that, if we look at the blood, we are starting to get to that population scale, but for a lot of other tissues, even if we just look at the single-cell RNA-seq data, there's still some ground to cover. So maybe one of the questions that we can reflect on in this session is why are we still missing some cell phenotypes? For example, does this have to do more with how we collect the data, how we collect the samples, or are there simply certain areas that we haven't explored yet? And can we use data-driven approaches to find which areas are missing? But also in terms of molecular modalities and spatial modalities, which cells or sample actually require these additional modality, and where will we have the maximum information gain from adding these modalities? The second feature that we'd like to reflect about is essentially we need the atlas to be stable. So a lot of the work that we do in computational analysis of single-cell genomics data now is very relative to the data that we're looking at. We do things like clustering, calculating cell-to-cell distances and KNN graphs. And these are very inherently data-dependent representations. But if we think of an atlas that has to essentially go through the ages and be updatable from version 1 to version 2, etc., we essentially need to move to stable representations. And also stable representation can take different views. So on one side, we could think of things like ontologies as stable representations. So if we have essentially a stable label for a cell type that we can expand and that we can relate to things like the tissues and the biomarkers, then this is something that the underlying data can change, but we can update this representation. But on the other hand, also when we were thinking about models and foundational models, this could also be a view of a stable representation that can be updated and doesn't depend on the data. But there's also, of course, trade-offs here in terms of interpretable representation, comprehensive representation, so we could reflect in this session about how to use essentially both views to create representation that are both stable and expandable for future versions. And also, how do we push adherence to standards? So for example, if we require people to annotate their cells but also add cell ontology labels, how do we actually make sure that the community does it? And as a third feature, the atlas has to essentially be useful. And for it to be useful, it first of all needs to be usable. So one of the sort of key aspects here is essentially interoperability. And here I wanted to highlight the work for some of the community efforts towards, for example, using data standards such as the data mu data objects that enable use of many different tools on top of them, but also essentially to have a stable structure to work with these single-cell data sets. But interoperability also means interoperability with other types of knowledge resources that might be used, for example, more by clinicians or by other communities that might want to use our data, but also task-specific optimization. So we've sort of mentioned that we're going to use the atlas for several different like high-level applications, but in practice, what we are going to use the atlas for also should guide how we build it. So what other uses beyond essentially cell type annotations and defining marker genes can we look at? We, for example, recently worked on how can you use the atlas for the task of identifying disease-associated cells. And there's features of the data that could be essentially infused in our integrations to enable different kinds of tasks. And so reflecting on how we do this could be another topic of discussion here. And with that, I think we can start by looking at some concrete examples of user stories and atlas integrations with Katy first.
Yeah, so we thought we could give you a few initial user stories and then see what you are optimizing your atlas for. And that could be a really, really interesting discussion, because if we could agree on a few use cases that we all are passionate about, then we could align many of the good forces that exist here across the community. So for the Human Reference Atlas, we have been discussing different use cases, including using the atlas spatial data, where the data came from, and if you have enough data and tissues section registered there, then we can actually start not just predicting what cell types should be there, but also we can guide cell type annotation. Or if we have a spatial location, then we can, again, using existing data, predict what cell by gene matrices might be there, which kind of biomarkers might exist there, and what expression levels might exist there. We also have implemented in many different projects are now using the exploration user interface, and I will have an example of that. And then going down to the single cell level, we are very, very interested in cell neighborhoods and functional tissue units, as we call them, and to really understand what happens when cells work together as a team, not just a single cell, but really looking at clusters of cells that have a very specific function to fulfill. And then it goes on to see ultimately how we can even have a clinical impact by, for instance, bringing some of the single cell data into pathologists' hands so that they can make better decision making. So on the next slide, and I guess I click here to expand. You see the exploration user interface, which now registers data from more than 6,000 data sets. And so you can now spatially explore that data. You can register your own data spatially, and you can see if some other healthy data exists for that specific location in 3D across 30 plus organs for male and female bodies. And on the right-hand side here, you would get to see the kind of data that exists, either as bulk data, as spatial data, or other types of data. And ultimately, you can click on one of these data sets and go over to the Vitesse browser to explore really that data at the biomolecular level. Very, very soon, the Functional Tissue Unit Explorer will become available. This work was very much inspired by the anatomograms that the EBL team brought together, but also the KPMP team has on their website portal the FTU Explorer, as I would call it. And these both teams have tried hard to segment out functional tissue units and then to display not just gene by cell matrices, but also protein by cell type matrices or even lipid biomarkers for single cells, or here in this case, functional tissue units. And just if you have data from, for instance, different age groups in the lower right, you can select those and you can see what happens to these matrices as we age. And if you have healthy versus disease, you could compare and contrast these different types of expression matrices. All right, and this gets us over to Graham's presentation, and you will be amazed to see what they just did.
All right. Hi, everyone. I'm Graham. I'm from Genentech. And I was asked to talk about the integration pipelines that we use in Genentech. And so I guess first kind of some basics and a disclaimer. We're a large company, large computational arm. There is no single integration approach that we take. And I mean, just like the people in this room, I think we all have a lot of different goals, and each one of those takes different integration strategies. So instead of talking about what Genentech does, I'm going to talk about what I do, because why not? And so I'm going to get into that. But one kind of definitional term, I guess, we use integration a lot. And to me, it's a fairly high-level term. It just means that we want to be using different studies together. And so that could be putting gene expression matrices into the same gene space, make sure that they're the same units, that type of thing. Whether you do batch correction or not, those types of questions depend on your application. And so integration just at the most basic level is just being able to use these data sets together. That's what it means to me. Okay, so I work on a tool called SCimilarity, so I'm going to talk about that. So to me, I think one of the key things about integrating single-cell genomics data, single-cell RNA-seq data, is the rapid growth and proliferation of this data. So as we track data sets, we can see that it's doubling. The number of studies, also the number of cells, is doubling about every 6 months. And so to me, when I think about integration, what I want to do is I want to think about integration of all of these studies. How can we put all of these studies to work? So it's not just a few studies at a time, but really as this curve grows, and in another 6 months and then 6 months beyond that, kind of how can we keep up with that? And so that's where I think representation learning comes in. We can develop representations using machine learning to do all kinds of things. Machine learning is a rich field, and there happens to be one subset of this called metric learning. And the special thing about metric learning is you can design these algorithms, these models, to relate cells to each other. And so we can take a look at all the cells in all these studies, and we can start thinking about how to relate them across these data sets. So this technique, metric learning, is borrowed from facial recognition, and you can take these examples of images of people, and what the model is trying to do is trying to place pictures of the same people closer together than pictures of different people. And so you feed it these triplets over and over and over again, and it's learning these features of the images that place them in latent space based on kind of this ordering or these similarities that they should have. And this is unusual in the machine learning field, or what makes this special is that we're actually learning to relate these images or data points to each other, and we're not trying to necessarily classify one image at a time. So we thought we could do the same thing for cells. We could take cells of different types, cells of the same type, and try to give them the same treatment, so that cells of the same type are closer together, and cells of different types are further apart. And so now we're building this representation for single-cell gene expression. And the idea here is now we're relating cells to each other. And so their placement, relative to the other cells starts to mean something about them. And so the idea is this should reflect some measure of similarity. The closer the cells are, the more similar they are. The further apart, the more distant they are. And so, I mean, that's pretty much it. I'm not going into the details in this talk, but I do think it leads to some pretty cool things that you can do. And so, for example, on one hand, we can take one query cell and we can compare it to a giant reference of about 23 million cells, and we can search to see, have I ever seen a cell that looked like this before? And so now, within well under a second, a 20th of a second, you can start to find other similar cells that look like this just by asking, just by representing it with our model and asking kind of who are the nearest neighbors. And so we can find all these different studies that are relevant to our cell of interest. And then you can dive in and you can do your regular classic analyses on each one of these studies to find which cells are the most relevant. And each study comes with its own metadata, the disease, the tissue, maybe some gene knockout or another perturbation. And so all of that provides context back to your original cell. And you can do all kinds of things. We can just visualize the data in one common space, and just so much that we can do from here. So this is the integration pipeline that I like to use. We developed called SCimilarity, and hopefully we're posting it by our archive like any hour, any day now. I wanted it done before here, but that didn't happen. So, okay, that is the Genentech SCimilarity pipeline.
All right, cool. Thank you very much for the opportunity to talk about CELLxGENE. I know this is a session on integration pipelines, so I want to start by saying we don't do that. Oops, I will go back though. So what we are working on though, to think about Emma's introduction, is to try to help create a stable and extensible foundation so that all of you can run your integration experiments. What we're working on and what we've compiled so far is a large data set. None of these data are ours, so I want to start by saying thank you to all of you who have submitted data to CELLxGENE. This is all built through partnership with data submitters who spend time with our curators working to ensure that they have all of the pieces of metadata necessary to run a good integration. Currently, CELLxGENE contains about 45 million unique cells. There's 850 data sets, and we have 680 cell types annotated in CL. We're starting to get large amounts of data. You can see the 8 million blood cells, 14 million brain cells, and some of the other tissues are beginning to hit critical mass as well. We compile data across data modalities and across assay types. So you can see there's a large list of technology, although I will say that the majority of the data in CELLxGENE reflect the data that you're generating, and so it's a large amount of 10x droplet data. Curation is central to what we do in CELLxGENE. The purpose of compiling this resource and spending time curating it is we hope that it saves all of you time so that you're not having to mine through geo and understand what all of the specific annotations are, but rather you can look at the 45 million cells through APIs that we generate and pull out just the data you want or just the data that's important for your scientific question. So through collaboration with others in the HCA and founded on ontologies that are primarily developed at EBI and by the HCA, we've aligned on a set of standardized cell metadata that every cell in CELLxGENE carries. And so this includes basic things like species and tissue, but we also have age denoted in age ranges, any diseases ontologized using Mondo, and we're increasingly trying to get ethnicity data. Sorry, if you're interested in the schema, we have a big QR code and also there's links, so hopefully the slides will get circulated. One of the things that's really important to us is supporting the Human Cell Atlas. And so what we're showing in this chart is the fraction of data sets that we've been able to compile in CELLxGENE or are actively working on for HCA bionetworks in the first phase of the integration efforts. You can see that in some of the tissues we're doing quite well. We either have the data sets in CELLxGENE or in progress for all of blood, and we're doing well in retina. And there are other cases where there's still work to be done. Some of our curators are here. They work with the Lattice team out of Mike Cherry's group, and if you're interested in having conversations about compiling data sets for your integration, they'd be very eager to talk with you. Another thing that Emma said was we want it to be useful, and so one of the things that we try to do for anybody who submits data sets to CELLxGENE is to maximize the amount of use of those data. So we have a series of UIs that we've attached to those data sets. The first one, I don't think I have a laser pointer, is the CELLxGENE Explorer, where you can go in and you can understand individual cell phenotypes. We also have a browser where you can find any of the individual data sets or collections. And up in the top right corner is the gene expression viewer, which integrates all of the data sets across the CELLxGENE corpus and gives a consensus view of the gene expression of any gene in any cell type. One of the things, and I think this is the last slide I'll show, is that's really important to us is community integration. And so what we've been working hard on are APIs that take the data that we've compiled and make it very easy for any of you to use them. And so what I'm showing here is the census. It's an API that's powered by a collaboration with TileDB. And in brief, what it does is it takes the 45 million cells and makes them accessible through simple filters currently in Python and R soon to come that you can use to take a slice out and generate an AND data object or a single cell experiment object or similar. We're working on extensions where you can take data and put them directly into PyTorch or other machine learning languages. But the hope is that by curating those data and delivering them through these APIs, we can speed the kinds of integration experiments that this group are running to start to really understand what the right approaches are and the pluses and minuses. Who's next?
Yeah. Okay, cool, thanks. How do I, the green one? Thanks. Okay, cool. Okay, so I'm Michaela Mueller. I'm a PhD student at Malte Lucken's lab and Fabian Theis' lab, and I'm part of the HCA integration team. And I'm going to talk a little bit about the infrastructure that we use as part of the team and something that Malte also mentioned earlier this morning. Yeah. So I kind of borrowed the slide because I think this is a really nice overview of like the general workflow, the general like tasks that we aim to do to be able to build a version 1 of HCA reference atlases for specific organs or organ systems. And this is kind of also what we based our a more end-to-end pipeline on. And also to clarify, in this case, we're actually talking about data integration based on aligning different data sets and trying to remove batch effects to have like a unified representation of that data. Right, so just in general, the 3 big steps are data metadata, which contains data collection and also aggregating multiple metadata in a way that we can use them across the different data sets. Then the technical phase where we actually do the integration, trying to also decide on a method in a less biased way by trying multiple different methods related, built in the integration benchmark. And then finally, there are a couple of downstream tasks that are also a little bit more manual. So based on this idea, I wanted to kind of explain a little bit what the backend looks like, because since we have these multiple different organ systems and somewhat different questions, we still need something to standardize the parts that can be standardized, I would say. So there are certain steps that should be done or could be done uniformly, and then there are also many different parameters that you can tweak. And the goal of this end-to-end pipeline is to also together, while setting up the different standards of what a good HCA reference is, also kind of encoding that in a reproducible code so that we can actually apply this to the reference atlases that we build. The main challenges here are data harmonization, so basically making sure that you that all the metadata that you have available is also usable and comparable across data sets, but then also on the level of cell type annotations, which is also a pretty hard problem. The other thing is we want to be able to tailor our integration strategy to the specific organ system or to the specific biology of that system. The final challenge, I would say, is the scalability, because now that we want to scale to multiple millions of cells, this is not as trivial. So yeah, an end-to-end infrastructure can help this. The pipeline itself is supposed to be a fairly flexible and also modular pipeline that can be tailored to the different specifications that different organs need. So now I will continue talking about a couple different aspects of the infrastructure. I guess I forgot to mention that, I mean, this is kind of a snake-make pipeline, so it's, in terms of definition, it is like something that has a defined beginning and an end, and is reusable. So for the data loading part, what we needed was to be able to collect multiple data sets and merge them together, and we wanted to do that in a fairly unified and automated way. We relied to some degree on the CellxGENE data because of the nicely curated schema. But we also allow for to map like multiple different schemas to that. If there are data sets that are not a part of the CellxGENE, to be able to use those data sets. Using this module, we can kind of enforce different data selection criteria to decide whether there are data sets that we want to keep or remove. So that's like one of the bigger uses of this module. And another module is, of course, the heart of the whole integration pipeline is to have like this benchmark for choosing an appropriate integration method. I want to highlight again, there's like no one-size-fits-all approach for these, and this benchmark is supposed to help us be a little bit less biased, try a couple of different methods, but the thing is the way we run the methods should be, probably does not require a new script every time, so that is something that can be parallelized and scaled a lot better if in this type of pipeline. And the benchmark itself currently has 8 different integration methods, but this can be expanded easily, and 14 different metrics to kind of measure the biological effect preservation as well as the batch removal. An extension to this pipeline, we're also thinking of new metrics to kind of, better measure certain more nuanced biology in the data sets so that we can have like a better evaluation of our integration approach. Right. And finally, I wanted to also highlight the aspect of tailoring to different atlases. So also in terms of usability, so we tried to kind of make a usable, I would say, kind of interface so that users can define what their input is and define the different steps, and also kind of tailor it. Also, you can in which order you want to apply which steps. And the idea is to in a human-readable format, have like all of the different parameters that you want to be using, specify them, and then that should be the only input that you would need to give the pipeline. Yeah. So thank you very much.
From recess. Yeah. So we would like to invite everybody who just presented to sit up front, and then we can start answering some of the questions that are on the last slide, I think. And I think Graham has to go to another event. So if you have questions for him, especially, try to ask them early. Oh, there are more. Now we have a lot of microphones. That's good.
So we can start by opening for questions from the audience for any of the speakers, for all of the speakers, or more general questions. We already have some hands here. I hope there's some handheld microphones left in the room after we have all of them.
Yes, there are. Perfect.
Sorry. So I'll be first, Fabian. So you heard a lot, especially in the introduction, which is lovely, from you, Katy and Emma, about the importance of optimizing approaches for specific biological questions. But there's an inherent tension between generalizable pipelines and wanting to have something that's quite bespoke for a particular question. So how do we, as a field, trade off that? Because actually the approaches that we heard about are actually pretty general rather than specific in terms of what they are getting at. So I'm curious how the people on the stage think about that tension between answering a specific question with a computational approach and coming up with an approach that's generalizable to other questions, which from a reviewer perspective, from a journal perspective, is actually kind of what we want as well. So how do we square that circle between specificity to a particular question and generalizability?
And I can start, and I think we can learn a lot from cartography, where for long times you didn't have a clock, so you couldn't do a latitude-longitude system. You had to do sight lines, and that worked to certain degrees. But then over time, the latitude-longitude system became an option, and it's widely used. We might not know the latitude, longitude, location for this room, but we all found here because we are now using additional value-added services that use, obviously, the coordinate system to get us to certain places. You also know that there are now many, many different apps which are customized for you getting your next coffee or your next mate or your next other item that you need in a geolocated way. And I could imagine that there might be 1 or 2 different common coordinate frameworks which are optimized for different purposes. But then the value-added services and the user interfaces will be very much customized for the use cases. And I think as soon as we could agree on what the common coordinate framework has to accomplish and agree on maybe 3 to 5 key use cases that we can all align with, then that would really propel us forward. And then there will be hundreds of other use cases that also will benefit. But there might be other comments on this. I think it's a great question. And it's always a tension. Whenever you make a map, you have that tension.
Yeah, I would also like to address this because I've also got heard a lot of comments of, I think, also different interpretations of what pipeline means. So I would maybe also clarify a little bit that, of course, a pipeline is kind of defined in a way that you have a defined input and a defined output, and you can make it a little bit more complex as well. For the purposes of the integration team, I would say what we were trying to do is there are certain steps that we know if we run a certain method, for example, right? We would run it, probably want to run it in a similar way. Yeah, of course, also being able to specify different parameters, but we still want to, the input and output of that task is fairly clear and fairly well defined. So I would say there are quite a few steps that have like a clear input-output definition that you want to make sure you want to do in a similar way also across different methods to make sure that the parts that are not supposed to change kind of stays the same. But then for the final atlas building itself, what you want to do is probably try out a couple of different approaches and then make a decision on how to proceed. So the pipeline itself is not supposed to prescribe exactly which steps are going to be done which way. It's more supposed to give you an option of multiple different, it's more like a toolkit, right? Multiple different options, different outputs that you can look into to kind of make decisions on your data and then at your data set, and then you can choose what exactly the final integration strategy should look like. So I think that would be kind of my opinion on the view on pipelines.
I can comment on this too. I think my view is that as we get better at training larger models, we can, and as we get more kind of experience in this field with machine learning techniques, we can train models that are for more specific tasks, very easy, like easier, but we also have a larger scale of data to use. So the models can be broad in biological scope, and I think can be, it's okay if they're narrower in kind of question, in application. For example, SCimilarity I think is quite general, but it actually is limited because it doesn't have any spatial data. So if you wanted to ask a spatial question, this is not the model for you. So I think models, I wouldn't go as far to say one model per question, but one model per few questions. But I hope that we're not retraining models every time we want to go and ask that same question in a different system, let's say.
I think if there's follow-up questions on this, Fabian had his hand up earlier.
And then Bruce.
Yeah, thanks for doing that discussion, and really liked also I think the point of the sort of Google Maps equivalent, we're sometimes overstretching it, right? Because you guys want to integrate, and I think many of us want to integrate across many people. So, if I was to take Mars's mind and I guess some other parts of liver, like pancreas, I guess maybe the overall shape might be a bit there, and that tells us, I guess, also about my naivety. But I think like local things will be definitely looking different. It's not all the brain where I think sort of these locations, what you just mentioned, Katy, we can sort of directly map upon. So I'm kind of really wondering what the way maybe to communicate, maybe also user interface, but before also to integrate locally would be that we would be describing sort of these local reoccurring tissue structures. In lung you have these sort of branching things. I guess you could maybe sort of a line along there, but then maybe you need to sort of define it along modules. So I think you call this FTU, functional tissue units. There were a lot of abbreviations on that slide. And then you had a CT times B matrix, which I also didn't quite understand. But would that be sort of an equivalent, if we had maybe, let's say, a local niche description, that's how I usually call it, then Graham, we could sort of chuck it into a big model again. If we could sort of agree, and at some point, we're going to be all drowning in this spatially, and we can sort of learn those large things again. But do we know that language? Is there already something that locally would be used for that? Or?
Great question again. So we have been looking out for these local niches, as you call it, local cell neighborhoods or hierarchical cell neighborhoods, as John Hickey and their team call it, and I think also the kidney experts here in the room, or functional tissue units, as others call it. I think there is value in looking at the functional units that actually have a very well-defined function and that exists in many cases a million times. Like in each kidney, you have these glomeruli, and you actually have hierarchical FTUs in the kidney that have been characterized by the kidney experts. So if you look at those and look at what happens to them as we age or as disease comes in, I think that's valuable. And we actually do have enough data for this, because within HubMap, in each tissue section, there might be 100 to 200 of those functional tissue units, and it's not really, it would be ideal to have 3D data, because they really are an interplay between the vasculature and other liquids that, for instance, in the kidney have to be filtered. So I think 3D data is absolutely essential for really understanding that all. But then you could focus on those and then build it up. So again, in each kidney you would have millions, 1 million of those. And then it's interesting to then see how that changes as we look to male and female, across ethnicities, across ages. And I think that's something which we can then also start to model. And I deeply believe that functional models would actually help us with this structure. I would always have assumed that we would go from models to get to structure and not to do an inventory and then try to get to function. I would, or at least do an interplay between the two. But that might all not yet answer your question. And maybe the question was, how do you really molecularly register a tissue block? Because that seems to be very hard for many that are surgeons or anatomists. They take a 10 centimeter kidney and now they are supposed to register a kind of small section of tissues that came out using a biopsy. And sometimes there are instrument readouts which can be used. And Ed Lane just showed this morning how they can take a brain from a real-life person and map it to a reference and the other way around. I think that's very doable. And so we can do this similarly, but then ultimately, how do you get it down to that very, very small scale level? And trying to deal with that multi-scale nature of the human body is really key to all of it. Would love to talk more about this, but I don't want to.
So can I ask a practical question about this? So what you're saying is that how far are we from, adding a column in cell by gene collection that adds to each cell functional tissue unit? Is this annotation always coming from the type of sample that you collected, or can it be inferred from the gene expression, or is this like tissue dependent?
Yeah.
Essentially, like, what's the state of the art for assigning from a single cell data set to a functional tissue unit?
So if you can register your 3D tissue or even a slice, 5-micron slice of it, using the registration user interface, which is freely available, then you know what anatomical structures collide, and through the anatomical structure cell type biomarker table, you know what cell types are commonly found in there. So if the biopsy went too deep and you have other tissue in there, you will see this immediately that something went wrong. So it's a great QA/QC. But you can also then take tissue blocks which are in cell by gene, and you can start to predict corridors from which that tissue must have come. And then you could say, all right, if it comes from this corridor, and maybe the surgeon still remembers it came from the upper lobe of an organ, then you could say, all right, that's probably where it came from. And this way we have at least the very critical information where spatially something came from. And I realize that in some cases you have organs that came to you in bits and pieces, and then maybe you can't register it anymore. But I think there is a way to then predict where it might have come from if we have some data that's spatially registered. And I think if you want an atlas, you have to have some of this data. Otherwise, you will never be able to project it back.
Okay. Data
So I think Bruce
Yeah.
was first, and then there's a lady in blonde.
We have to wait for the mic for the online.
So many questions. Katy, I mean, I loved your cartography example, but I think in what you last said right here, you're way underestimating the problem of going from XYZ coordinates with such and such a neighborhood tag to actual what's the structural unit that we're looking at here. So, you know, I can say, okay, what's the typical architecture of a, you know, of a 2-lane by 3-lane intersection with, that only has the, you know, that has a left lane turn lane on it or not, and then once you kind of say, okay, this kind of a intersection now, we know the general architecture of what needs to be there and what are the elements of it, and then here's this instance of it that I'm looking at, which is, you know, that corner right out there, you know, at Mars Discovery District, and it, you know, and it conforms to this rule. But behind all of that, those concepts is a very complex data model that talks about all these different features and their relationship to each other. We are so far from that right now. So, you know, I think that this is, you know, when you talk about like subcellular spatial genomics data right now, you're really talking about, you know, XYZ position, and you know, you're starting to segment the, you know, the elements of the cell and where, and then what's its, you know, the patterns of the different cell processes, and that's all in the context of, you know, where's the basement membrane and where's the vascular element, and then which cell interface at, you know, apical side is there to what's the subdifferentiated state of this other cell on the, you know, with laterality on it. All of those things are going to, you know, ultimately going to be critical to really understand FTUs. And, you know, I looked across the hallway at Ed, and I don't know if Ed could read my mind, but this is all stuff that Ramon Cajal was thinking about, and, you know, and then just the genius of the representation of those, you know, kind of the features of very complex structures in, you know, in the brain. So can we learn from that? How about, you know, completely outside of you know, the data collection stuff right now, could we come up with frameworks, knowledge frameworks that really harvest all of our priors from all the Ramon Cajals that, you know, have ever put diagrams into manuscripts that say, here's the typical tissue architecture of blah. You know, could we, you know, go through and then do a, you know, kind of an image analysis for best cartoons, you know, of XYZ tissue areas, and, you know, and the way that people schematize things in, you know, in very kind of bespoke situations, I think that's all harvestable. I think that, and that's the framework that we have to kind of to build on for real, you know, understanding of, is this, you know, is this a place I can make a left turn onto this kind of a street?
So we have had experts agree on 20 of these functional tissue units. So they're all from different organs, and it's not easy because it's spatially explicit. It has to have all these cell types that ultimately are linking to the cell ontology and Uberon IDs. And ideally, we would have them in 3D. But given that we have them in 2D, we can now start comparing these human-generated 2D functional tissue units with what comes from the data, right? So that we basically, he's allowed to do this.
I did have a question for you, though. So, like, in thinking about, so it's a data model problem. I just want to say, like, when you see problems in single-cell data sets that they're not annotated properly, and you see, as you harvest single-cell signatures, they're not annotated properly, and, you know, and Malti and Lisa did an incredible job of doing the error type analysis of what, you know, what kind of a classification situation is this on, you know, from the original data to the transformed data. And there's a ton of badly annotated data in our databases right now. What's our approach to, how much are you seeing that is one thing. And then the other is, how are we correcting for it in, you know, kind of a secondary ingestion pipeline stuff?
Can I steal it? I'm going to steal your mic. We re-annotate everything, actually. I mean, not just for quote-unquote wrong annotations, but we want a common definition. If we're going to actually use these data sets together, then we all need to agree on what the difference between a monocyte and a macrophage is, you know, across these data sets. So,
how massive are some of your corrections?
That's a good question. I mean, for the most part, I think people are pretty good. I think what we see actually the most is Well, and what really sticks out quite clearly is, if there's like an off by one error, then the whole data set doesn't make any sense. And so that is actually quite visible. I think, I don't know, I haven't gone back and kind of quantified at a study level. I mean, that gets down to names and, no, just kidding. Yes, yeah. But I do want to emphasize the importance of the common definition, even besides just trying to correct mistakes. Even if everyone did a great job, which I think a lot of people really do, you still need to, if you want to pull out all the macrophages from all the studies, we need to agree, you need to have one definition for what that means to pull them out. So, yeah, I think reannotation is actually, I strongly advocate for it. Okay, I do have to run. Okay. Sorry, guys.
I'll release you, Graham. So this is a really interesting question for us at CellxGENE. If you'll forgive me. Sorry, I know there are lots of questions. And we don't have a clear answer for it, so this would be something we would love to speak with you all about. We are running an experiment where we ask everyone to map all of their cell type annotations into the cell ontology. And our understanding is that for the most part that works really well. Every now and again there's a term that isn't there that you'd like to have, lacking specificity. And then we get all these different data sets that have been annotated by you experts, and we can run reanalyses. We can map them all into the same space, and we can ask, why are the labels on these two cells whose gene expression or distance in gene expression space is very small, why do they carry wildly different labels? We've run one analysis recently which revealed one thing that I think is interesting that I'll just throw out there, and it's that sometimes there are just differences in salience or attention. Somebody will submit a data set that focuses on, let's say, the lung, and they may not care so much about some of the cell types, and they're not annotating them incorrectly, but what we see is that they annotate them at a low level of specificity. And so those cell types then functionally get lost in all of these reanalyses or queries because somebody might want to find a really specific type of immune cell, but they've been annotated lymphoid cell or something like that. And so that's one example of an opportunity when we comb through our database that we may be able to more specifically annotate these cells in future passes. How we go about actually doing that and going back to the authors and saying, hey, we have some evidence that suggests maybe this could be improved upon is one opportunity, but then we'd have to come and talk to you all the time, and I don't know if you all want that. So, interesting open question.
Chloe's dying to ask a question.
Okay, Sam, is it, because Sam manages the virtual event, but if it's not related to that, I definitely want to pick up on that.
It was partly related to this. It was more about how we've got a question from the virtual audience. Some of the integration, it was about annotation, some of the integration benchmarks rely on the cell type annotation. But how can we avoid being circular on annotating data and then using those annotations to decide which integration method works best?
If I may, because that's probably related to our workflow. Yes, this indeed could be a problem, right? The thing is, for the integration, I think to a certain degree you have to go with what you have, right? One option, of course, would be to go into every data set and then annotate it super cleanly, re-annotate it super cleanly, and then do your integration. But you could also go with a somewhat more time-saving way of trying to leverage the author annotation and the assumption that they're to some degree correct. They don't have to be perfect, but they should to some level describe the biology you're seeing. So usually if you go to a lower cell type resolution, it is more likely that the cell type annotations are correct than if you go to something very granular. But what we try to do is kind of leverage as much information as we can, assuming that these author annotations are to some degree correct, and then just to get a general ranking of the different methods. After that, of course, you need to look into your data sets and then verify if it really makes sense or if you're biasing yourself to a certain metric or not. So that is not to be excluded, but this benchmarking and then having a ranking is already a huge help because then you can already see, reduce the list of integrations you want to look into. Yeah. Well told. Add something to that.
Chloe was
well, it's really too bad that Graham is not here, but I'm still gonna, I wanted to kind of tackle back to what he was saying back to Bruce and just more generally speaking. And so I think this is another example of tensions, perhaps between some of the computational frameworks, repository, and domain experts that are annotating cells. And it often makes me, I will just say, speaking on my own name, although I know a lot of bionetwork members feel the same way, makes us cringe when we hear that some computational biologists have relabeled the cells that in some case we spend months and years annotating. And then they come and don't necessarily engage with us and then decide that this is the common label. But I can appreciate that there's actually a need to define common structure, to define this benchmark. And so how do we navigate that tension? How can we define a common lexicon to figure out that, okay, Bruce, I'm picking on you. Maybe you and I are not defining, we're not giving the same name, but actually it's the same cell. We've been talking about the same cell all along. We just have not been using the same features to define it. And how can we proceed with our integration efforts without ignoring months and years of work? I recognize that some data sets are not well labeled, but some are. And unless you have really expert knowledge to figure out which is which, you can really much bias the integration. And this is a part where we really need to have tools for the whole community to engage in that conversation. So that at the end of the day, we create a product that everyone will adopt, and not just a handful. And so this was the impetus to create the Cell Annotation platform, which is a platform for the Humans Atlas. It's really meant for this community, where we can accumulate everyone's justification for calling a cell a certain way. What were your justification? What were your proof? What were your evidence? What were your synonyms? What's the relationship? And so if we can do this for every data sets, with Michaela did a beautiful overview of the framework she's presenting, which actually is needed. But then we can actually interpret the results of our integration by tying it to the annotation that originally provided. And then as a community, we can figure out which one are supposed to be the same, and then together decide how we want to call it. And then, of course, update the CELLxGENE and other repository with these labels with our justification. But I find it, sorry, I know it's not a question, it's a comment, but it's a slippery slope to relabel cells without engaging the wider community and without understanding the justification.
If I may, I would also add to that point. So I would say in general, the whole process of atlas building is also very iterative. Another reason why we might want to have a pipeline to kind of be able to repeat these analyses with somewhat different settings. So I would say if your first pass or your first set of labels that you agree is to some level consistent across data sets, that might not be perfect, but that's still better than not having any labels at all. And you can already use that to do your integration.
A microphone is coming to Malte.
Keep going. Oh, sorry. And then once you kind of look into integrated embedding, right, if you decide, well, we're trying to re-annotate, also with expert knowledge, with the CAP, the annotation jamboree, trying to involve the community as much as possible. If you do find that things don't fit, that it's nothing speaks against reiterating a couple of steps, going back maybe and deciding if there's something obviously going wrong to go through that process again.
Yeah, I agree. And I know Mata is, really wants to jump in. It's just I do want to recognize Michaela's beautiful work. There's a distinction between what you're doing, which is try to benchmark your integration effort and just finding broad commonalities. And it's too bad that Graham ran away. It's different from what he was doing.
So maybe I can add very briefly. It's really nice, Bruce, that you're giving Lisa and I a lot of credit for identifying misannotations. But really, these were 6 bionetwork experts who gave their input on every single annotation, and we collated those to get a majority vote. So really, the bionetwork experts re-annotated the same cells that they had previously annotated again in a consensus fashion, and thereby you could find these kind of tensions between the original annotations and maybe the new annotations. We went back to all those experts and they all agreed with this final annotation. So it's not really, I think it's really important to highlight that this is not a case of a computational person coming in at the end and saying, oh, by the way, you all annotated this wrongly. And what Ambrose said before about the hierarchy of annotation, that was a very strong factor. I think about 40% of cells, which we initially called under-annotated in the lung, was just because as Ambrose mentioned, this was not the focus of that particular study. If you're focusing on epithelial cells, why are you spending a lot of time on endothelial cell subdivision? And this is, I think, what happened a lot in the field, because this was just the target, the goals of those papers. And then bringing that back to the kind of cell label encoding, what we're trying to do that is kind of just bring a common denominator in a non-contentious fashion, to respond to the online question as well. This is not the idea is that this could bias, but we've actually found previously that we can relabel a lot of cells with the community better than this initial label encoding, even though we are initially selecting methods in this way, using that label encoding. So it doesn't have as strong an effect as you might think.
I also wanted to highlight that Malte this morning was also mentioning methods for essentially finding synonyms. So harmonizing labels without having to relabel. And so maybe a shift in thought here is not just that you don't need to put a new name, you just need to put all the names that are related to each other in the same bucket. And yeah, just wanted to, ongoing computational work on this, I think, is very promising.
And reporting from the trenches together with Malte, Matthias Kretzler from HubMap, KPMP, and Human Cell Atlas, and you can see with varying 3 or 4 heads where the complexity already is coming from. This is really critical. And we have to be practical here in the sense that, yes, where we can build a clear community consensus, that should be represented. But I love the cup philosophy because it allows us to let multiple flowers bloom. And scientists are scientists. And if you force scientists into a rigorous structure, they will not operate in that. And we end up with a Tower of Babel where simply a lot of our knowledge is not mappable. So having the CAP framework in place where you can project the different data sets onto each other and not forcing people to a top-down nomenclature consensus will be critical for success of our community. Otherwise, we will not understand each other. And we know where Babel went. So please, find the right balance here. I think HCA and HubMap, and thanks God, Katy is active in both of these frameworks, and with the ASTC plus B tables, we have a very good basic knowledge framework emerging, that we have that available, but let's keep flexibility open for people to stick to their own strategy, but still be linkable going forward.
I think there was a question for Kirsten.
I'm Kirsten Meyer from the Sanger Institute in Cambridge. So I just wanted to follow up on Emma's point. That there are now very powerful tools, and one that I wanted to plug in a way in the absence of Chuan is actually CellTypist version 2, which is very powerful at identifying cells that are identical, even if they have separate labels. And I know in the early days of the lung integration, aligning things also between Human Cell Atlas and Lung Map, it is hugely powerful to get a consensus to find that 2 cell types are actually the same, but just labeled differently. And CellTypist 2 is really a very powerful tool to do this, and it'll also allow you to understand which cell type is a subcluster of another cell type. So it provides you with a hierarchy, which again is hugely helpful in getting labels, because sometimes people agree that it's part of a particular larger, broader term, but then have more specific subterms that an earlier study might not have had. So yeah, I encourage people to try the tool out.
Just also mentioning there is spatially explicit data, and for that it's very common to have a human-in-the-loop approach, where you basically have the machine suggest certain types of segment.
May I?
So you have human-in-the-loop approaches where the machine does all the dirty work, if you wish, and then experts would correct it. And then this collaboration between human and machine intelligence is actually making both types way happier, because the machines learn from the corrections they get from the human users, and the human users, they don't have to single-handedly outline every single cell or every single FTU or every single anatomical structure that you want segmented, but it can be much more automatized and speeding up the process. So I think that human-in-the-loop approach is very powerful, and you might like to have something like this also for cell type annotation for bulk data.
We have someone here who's holding the microphone for ages.
Yeah, thanks. Look, Well, my name is Thiago. I work with the cell ontology and other ontologies organizing information. And a lot of the points here are very interesting because when we work with this formal organization, a lot of these specificities that are important for describing the cell are eventually lost. And different communities have different ways of organizing that, right? I mean, on taxonomy, you have a framework where you can say how do you describe a species, and then there's a nomenclature committee that decides if the names are right or not. And then you have GNC for genes that define the names. And for cell types, we kind of have the cell ontology that has been going on for 20 years, but it has different aspects. And for anatomical things, we have Uberon and other anatomic ontologies. But it's an environment that can ever change, and I think that in many ways it doesn't fit what the community needs. I mean, sometimes you need better granular annotations, you need some degree of expert things. So I want to just ask you, in this world of ontologies, controlled vocabularies, or other types of organized ways of putting data, what is missing? What is missing out there that we need to build? Which kind of resources, ontologies, or governance structures for updating these resources we need to actually cover all this diversity in cell types and cell functions, and etc.
Well, I'm just gonna, build upon what you were just mentioning, and also one of the points that was made earlier that we can come up with the best standards possible, but if we can't get people to adhere to them because they, maybe work in a different ways, then they're not useful at all. So I think, for example, when I've tried to use cell by gene, I've used a lot cell by gene datasets because they have the cell ontology, and that helps. For example, in putting together datasets without having to do an integration where you have to put everything in the same latent embedding. If you're interested in lineages, you can go higher in the hierarchy pretty easily, and you can use this information. But then, and so it's great that the collections have this essentially requirement to put the ontology label on top of your own label. But then in some dataset, people are sneaky and they just call their cell. So they use the ontology for a native cell. And so that essentially, you have to, how do you essentially encourage people to be, to be mindful to the community in this sense, or why is this matching hard so that you can't really do essentially the rename with the cell labels that you gave into the ontology? So I think a lot of it might be essentially putting what's already in the ontologies now through, for example, these pipelines to find synonyms to see where are we actually, we have no synonym in the cell ontology that we can use for cell types that are actually widely found across tissues or, across studies. I think that could already, maybe help a bit, because that ontology was also built not necessarily by the single-cell RNA-seq community. So we're taking the knowledge from the single-cell RNA-seq community into that, and these synonym methods could, I think, really help with that.
Yeah, if I could add, I think a reflection that I have is over the past year, I think the use of the ontologies in our space has become non-controversial, which I just wanted to say out loud and state my excitement for. But one thing I think we consistently hear is that the ontologies, because they need to be evidence-based, they lag behind the cutting edge of the field. And so if we accept that paradigm, and Matthias mentioned, this need to be able to, to be able to continue to expand outwards and do our own thing and then converge back to what we agree on once we've run all the experiments, then the question is, how do we keep the ontologies up to date or as close to the cutting edge as we can, and how do we balance accuracy and agreement against experimentation? And I think that there are some really cool community initiatives there. I talked about an experiment that we ran very simply just to look at the differences in expression space. But I want to give the mic to Chloe, who probably has something relevant to say on this.
I just want to piggyback on the ontology because, to your point, it's really nice that it's getting non-contagious anymore, and it kind of, to Emma's point, it kind of can really help moving forward the field. And so I guess we didn't flag this because it's going to be discussed on Wednesday, but as part of the cell annotation platform, we recognized that we needed to also help update the ontology. And so, David, you're part of that team with David Estomie Startlands, and We are, as we accumulate everyone's annotation metadata, we also try to link it to existing ontology and identify gaps. And we briefly mentioned during panel discussion this morning cell annotation jamborees that will take place for all bio-networks. As part of this, there's going to be, these are going to be done also in collaboration with CZI, CellxGENE's team. And so as part of this, there's going to be an in-person component, a big virtual component to give everyone the time to do their homeworks, but an in-person component to come together and discuss agreement and disagreement in an annotation with the goal and having members of the team that thinks deeply about ontology so that by the end of the meeting we can update the ontology for every particular system. And it's not going to be perfect. I think, Emma, you were saying this earlier. It's going to take many iterations, but that's kind of being mapped out and supported just to kind of flag it because we recognize that's important. And we want to make sure that people just don't label their cells. Yeah, just want to flag that the leadership has recognized that and there's some structure in place to help support the growth and to linking it to different repository.
And another very systematic way of getting your new cell types and also anatomical structures, niches, cell neighborhoods that you have identified into the existing ontologies is to just add them to these anatomical structures, cell types, biomarkers tables, and then as soon as David Osume-Sutherland's team sees that there's a new one, there's an issue tracker, and then it gets discussed, and slowly and steadily, it might take 2 years to get these 5,000 new terms that are needed to capture healthy human beings integrated. It's a huge ask for you guys to put so many in there. But I think right now we are forcing existing experimental data to align with cell types and Uberon terms, which might not yet capture in the existing ontologies what we see in the data. And that's also a little bit making me unhappy a little bit. So ideally there would be something that goes faster, but I know that this is a very systematic process to advance these ontologies.
Just to be explicit, our curators have learned the native cell trick. They will come back to you and they will talk to you again about that.
We have too. Oh, that's what you're addressing.
There's someone that hasn't talked yet, so.
Hi, sorry, I don't want to that we talk all about annotation and non-integration, but I have a question / a reflection. So I think it would be somewhat easy to involve everyone that is participating to the HCA and come to a somewhat agreement on annotation of cells. Oops. What I don't know, how we get the people that publish in the field, but for whatever reason, they're not involved with the HCA and they're not actively participating. Do we need to worry about people that keep publishing it almost in a parallel way and they have their own annotations and they really don't engage? Can the journals, for example, help us? Or maybe I'm wrong, maybe we don't need to worry. I'm just curious what you guys think.
Just one idea. I think there are opportunities for us to help ourselves. I think if we as a community are building resources describing cells with new levels of resolution and we put that out there and make it usable for the broader community, they'll begin to see that and they'll begin to interface with it. And if they see value in it, which we hope they do, that's one way that we can begin to draw them in so that we're not having to push out to them all the time, but rather we can capitalize on what we're all interested in doing.
I would also say, like, I think kind of what we're talking about right now is a little bit like new data generation, but like in the context of integration where you say, well, we want to use existing data and then integrate it together to come up with like a bigger representation, you know, one where you might find cell types that you couldn't before and so on. If that involves reannotation either way, I guess, I mean, depending on, it doesn't really matter which data set you get too much in the end, if you can kind of re-annotate it in the, I would say, yeah. So say if we have like a community effort to re-annotate, you have like an evidence, right, just on a single data set, but then if you go back in an integrated embedding and re-annotate and find that there might be more nuance in the cell types, I think that might also be a way to actually just collect that information.
We have another question. Yeah, someone with the microphone.
Great. Thank you for the discussions. And also shout out to the CellxGENE census. We use it a lot when we train SCGBD, so great work. My question is more or less related to some of the future work for CellxGENE. I think you list some of the question here is how do we expand the current scope more than just RNA-seq? Can we expand to, let's say, a taxic or spatial omic, or even, as you mentioned, can we link cell by gene to other resources such as HubMap? You know, what's the plan you have for expanding cell by gene?
Thanks for the question. So I'll answer it in two parts. One was about modalities, and the second was about cross-linking. On the modalities topic, we're pretty confident from conversations with many of you that the next thing we should add support for is spatial data. After that, I think we have some open questions, so if you have opinions, I'd love to hear them, as would others on the team. So come tell us what you're excited about. Cross-linking, I think, dovetails nicely with the rest of this conversation, because as the fields begin to converge on standard representations, it makes it really easy for us to use those entities to link across. And so we're looking forward to linkages to the Human Protein Atlas, to more linkages with Katy's team. And our vision would be that anyone who enters any of the resources being developed by the community could find all of the related resources and navigate based on gene entities or cell entities or diseases. So from that perspective, we're really excited about the standardization that we're seeing, because it's going to manifest as a more interoperable and useful ecosystem, we hope.
I think we are at time. So so yeah, thanks everyone for contributing this discussion and for the ideas. And yeah, I don't know what's next, and I hope someone comes and tells us.
I think there's a break coming up. So continue to enjoy each other's company and questions, answers out there, and come back next year. I think we will have a similar discussion still ongoing, but hopefully with more APIs that all nicely play together. Thank you.
It should, based on Graham's talk, be 300 million cells that Graham has. That's what he promised.
Very cool.